Diamond, the managing editor of Infection Control Today, where we're trying to give infection preventionists and other healthcare workers the information they need to care for patients during the COVID-19 pandemic. COVID comes equipped with words and phrases that now gets everybody's attention. Flattening the curve, herd immunity, N95 respirators, quarantine, social distancing, and vaccine. That last one, vaccine, especially gets a lot of attention. But until one is developed and distributed widely, another word is often said, testing. COVID-19 testing is always in the news, sometimes creating controversy. Are case counts really rising, or is it just a result of more testing? What about all the different kinds of tests that are out there and the reports of false negatives? And that brings me to today's guest, Dr. Jack Regan. Dr. Regan is the founder and CEO of a company called LexaGene, a company that makes COVID-19 tests. You can find out more about LexaGene by going to the company's website. Okay, but that's it now for the plugs. Dr. Regan has agreed to visit us today as an expert, not a CEO, and we'll talk to Infection Control Today about the challenges inherent in developing tests for COVID-19 in general. Dr. Regan, thanks for joining us.
Thank you so much for having me on the show. Appreciate it.
So let's start with a general question about COVID testing. What are the main challenges involved in developing such a test?
Well, I mean, COVID is a RNA pathogen, and with RNA pathogens, you have to obviously do two types of chemistry rather than a single chemistry. So typically, when you think about detecting the pathogen, often you only look for the DNA. In this case, COVID, caused by the SARS-CoV-2 pathogen, is an RNA pathogen. That means we have to convert the RNA to DNA first before we actually amplify it. And so there's an extra step involved in detection. It makes it a little bit more complex. It is a very complex chemistry. This is why generally this testing is best done in a certified reference lab by experts. And one of the big needs, which I'm sure we'll get to today, is automating this technology to make it faster and more accessible so we can actually bring it out of the reference lab more to the point of need where diagnostics are needed for fast turnaround time.
What kind of experts work on testing? Are we talking chemists or engineers of some sort? What expertise is used to develop tests?
So in a reference lab, there are people who are trained to be clinical laboratory specialists. These are people who go to school to learn the proper techniques of doing molecular biology effectively. And so in order to know those techniques, you have to be very careful so you don't contaminate a sample or assemble a test in a wrong fashion, which would lead to a false negative result. And so there is not only a science, but an art to it. And this is why, you know, many labs like to try to automate the process, because even though these clinical laboratory specialists are highly trained, we are human, we are prone to mistakes. And so there's obviously a need and a desire for automation. And automation effectively removes the human from the scenario, ideally generating higher quality data.
At what point do government overseers come into the process? Do you keep them informed every inch of the way of your development, or what?
So in human clinical diagnostics, the FDA does regulate testing. They do this to protect, obviously, the patient and ensure that a quality test is being administered that gives as accurate as a test as possible. However, no test is considered 100% accurate. It's just a matter of the nature. It's impossible to have 100% accuracy. Often you have dual competing technologies where they are very, very similar, but there's always discrepancy. And so regardless, the FDA does regulate it. They want to make sure there's a high standard of quality. And so for companies like LexaGene, we have to develop a data set, submit that data set to the FDA. They will review the data set. If it meets their expectations, and this means it has to be as good or better than a predicate device, then they would grant us the authority to sell into the human clinical diagnostic space.
Test somebody today, they walk out of their house, go mingle in a crowd, and by 3 days from now, they have COVID-19. What test can possibly guard against that kind of spread? COVID-19 is a fast spreading virus.
Right. I mean, it's really impossible to completely stop all spread in those circumstances. in regards to having somebody who just recently got exposed, the probability of testing them and actually detecting the virus is so infinitely small, generally they'll come back as a negative. That is because, say you walk by somebody who coughs, you inhale some aerosols, that aerosol contains, say, a coronavirus. It lodges deep in your lungs. You may have a couple hundred viral particles in your lungs, but it's deep in your lungs. If you do a nasal swab sample, the chances of you actually detecting that are close to zero. And so this is why there's an incubation period where the virus will infect the cells in the lower lung. Within those cells, the virus replicates, multiplies in numbers, and starts to spread. And as you breathe, the virus gets spread, throughout the rest of your lungs and ultimately up into the upper respiratory tract, where you can more easily detect it. This is just the nature of infectious disease. These viruses are trying to survive, and they do what they do. Our job is to provide high-quality, sensitive diagnostics that allow you to detect it earlier than you might with other technologies. So there are different technologies that are more sensitive than others. And so, PCR is extremely sensitive because if a copy of the coronavirus genome is in the sample, it goes through a massive amplification step. And if you look at PCR, and if you're a mathematician, basically in PCR, it's exponential growth over 40 cycles of the reaction. If you do the math, that translates into roughly a trillion-fold amplification. So PCR is considered the gold standard because of sensitivity. Ultimately, to get a handle on a virus like coronavirus, it's very, very important to slow the spread of the disease. Reduce that R naught factor down, so less people are getting infected from every single person who has the disease. And really, the only way to do that is quick, rapid diagnostics that are very sensitive. Identify those people, make sure they self-quarantine, and really limit the ability of the virus to spread to other people.
When a vaccine comes along, will that make testing irrelevant?
No. And so people think of a vaccine as a silver bullet. And unfortunately, in regards to viral RNA vaccines, first, a little history here. I did my doctoral work on influenza. I'm an influenza expert as well. And so the influenza vaccine is about 14 to, at best, generally 85, 90% effective from year to year. It goes up and down. You're looking for a vaccine which ideally is at least 70% effective. Here we are looking at coronavirus. It is a new class of virus. There are other coronaviruses that are less virulent than this SARS-CoV-2 coronavirus that's causing the current pandemic. That said, we've never developed a vaccine against coronavirus. And because of that, the question is how efficacious is the vaccine going to be? How long is the immunity going to last? If you've been reading the press and reading some research papers, what they're saying is that there's some patients who are getting exposed, they peak in the amount of antibodies that they have, and those wane after just several months. And this is very, very concerning because if the antibody level doesn't stay sustained for a long period of time, you could literally get either exposed or vaccinated and then get sick again, literally 2 or 3 months later, which is very, very troubling. And so, because of this, diagnostics is always going to play a big role. We should not be looking at this winter with the vaccine coming as the end of the pandemic. One, there's going to be a lot of issues with distributing the vaccine. We don't know how efficacious it's going to be. There's going to be a lot of different vendors providing vaccines. Generally, what ends up happening, unfortunately, with the pandemic is certainly we hope to flatten that curve and reduce the number of mortalities and people who have severe illness. But generally, they only, quote, sort of go away after herd immunity has been developed or the virus is just sort of, and that happens eventually. It's not like this virus is going to wipe out the human race. This virus will continue to evolve. Our immune systems will evolve. And ultimately, this will fade away, but it likely might be a full 18 months, if not longer, before that happens, which is very, very frightening.
What's the most effective vaccine that has been invented? I mean, I guess polio vaccine eliminated polio, so you have to put that in the running, right?
Right. There are vaccines that are extremely effective. Polio is an example. It just so happens that vaccines against influenza and coronavirus is an RNA virus similar to influenza, one segmented and negative strand, the other's positive sense in a single strand. But nonetheless, there's a lot of similarities between coronavirus and flu. And if we look at flu as a model rather than the polio vaccine, we expect a coronavirus to act more like flu. And the reasons for that are a couple reasons. One, when a pathogen is an RNA pathogen rather than a DNA pathogens are very stable. They don't change. Now, that is because when they're replicated, the fidelity of the replication machinery is very good, and so the pathogen doesn't change very much. In contrast, influenza, an RNA pathogen, like coronavirus, an RNA pathogen, the fidelity of actually replicating the genome is not good. And so you end up getting what's called a quasi-species. Getting a little bit technical here, my apologies. What a quasi-species is sort of like a cloud of viral particles that are all a little bit different from one another. This allows viruses that have a new inherent mutation. That mutation is either going to be good or bad for the life of the virus. And so obviously, with sort of natural selection, if you will, going against immune systems, these variants arise that evade the immune system and make it harder to actually develop a good vaccine for it. And so this is one of the challenges that we have with vaccines, diagnostics, and therapeutics with RNA pathogens, just because they're always changing. It's a moving target. And it's a challenge for all aspects of medical care.
Doesn't sound very promising for the development of a vaccine, but they're going for it anyway, as they should.
There's a lot of companies working on vaccines, and certainly they will provide value. I don't want to, discourage hope from people. I myself personally am very much looking forward to eventually receiving a vaccine. Vaccines are overall safe. There's a lot of anti-vaxxers out there, which I would argue to them to read the literature and understand the risks of vaccine usage, because the risk of not receiving a vaccine is far greater generally than ever receiving. We have to massively increase the amount of testing. I mean, it's one of the things, if you look at the countries that have served as a good model for testing, they were able to ramp very, very effectively. And so we need to do the same thing. We have to increase the amount of testing that we're doing. Right now, the FDA is looking at other countries, what they've done. They're starting to follow some of their guidelines. And one of the guidelines has been pooling samples. I mean, manufacturers are working as hard as we can to develop the test to allow for testing. But fundamentally, we've gone from a relatively low need for testing to a massive, massive need, where there's needs to really test arguably, 10 million people a day, if not more. And so to hit that capacity is very difficult. One of the things that the FDA is looking at right now, or has looked at and made recommendations, is the opportunity to pool samples. And so pooling samples means you take a lot of, say 5 or 10 different individuals who have submitted their sample for testing, you combine them into one, and you process that one sample on an instrument. In order to do that, you have to use a technology that's extremely sensitive. Vaccines, diagnostics, therapeutics, they all work together. They're not independent. And so what we hope is that the vaccine will provide some level of herd immunity. The other part of the herd immunity is going to be natural infection. The people who are sick are going to receive receive therapeutics and hopefully not die from the infection. Diagnostics are intended to curb the spread of the disease. We want to prevent people from getting infected. We want to identify those who are infected, stop the spread. The only way to do that is to identify early and with a sensitive technology, and to take behavioral changes to stop the spread. One of the things to remember is there's been some arguments by individuals in government that if we don't test, we'll have less cases. That in many ways is true, but you won't have less illnesses. If you don't test, the number of illnesses is going to expand significantly. If you look at the number of illnesses in the United States today, there's been roughly 3.5 million documented cases. There's some arguments to say that the number may be as much as 10 times higher than that. So people who weren't really severely ill or asymptomatic never got tested, right? So that puts that number at now, say arguably at the top end, 33 million, 35 million people have been exposed to this illness before in the past. If you look at the United States, I'm not sure the exact number, but I think roughly in the United States, there's 360 million people or so in the United States. We're at, roughly a tenth of the population has probably been exposed to this virus. If we do not test, if we do not develop a vaccine, what will happen? Generally speaking, to achieve that herd immunity, you need to have roughly 75, 80% of people who've already been exposed. That means we have arguably another 200 million people who will get infected by this virus. If you look at the mortality rate of this virus, depending on which numbers you read, and in regards to the people who have potentially been exposed but not classified as a true case, the mortality rate is somewhere between probably at the very, very low end, 0.25%, probably more closer to 0.5%. But if you just go strictly on the number of cases, it's closer to 4 or 5%, which is very, very frightening. That means we have many, many more deaths in front of us if we do not work as a concerted team between diagnostics, vaccines, and therapeutics. All three have to work together. Obviously, the pharma companies are working hard to develop therapeutics and the vaccine.
Imagine you're taking a look at what's going on in other countries. Does any country stand out to you as being doing testing the right way or better than other countries?
Well, certainly like South Korea and Australia, or I think it's New Zealand, has done exceptionally good job of containing the spread of the virus. And so those are sort of model countries which have done very, very well. China as well. Look at China. They had the outbreak initially. They did a massive, massive effort to get that under control. And in the Asian countries, there are more used to these viral spreads. If you look historically at a lot of the pathogens that have emerged, they were much more willing to adopt wearing masks. Unfortunately, in the United States, there's many people who do not want to wear masks. It's something we've never had to deal with before in the past. It's always been effectively a problem that was sort of more associated with the Asian countries, but that's completely not true. The virus doesn't know any borders. And so the countries which have had past experiences with outbreaks, like SARS, like MERS, et cetera, avian influenza, they were much faster and aggressive in their rollout of testing and really mandating self-quarantine. The minute people start doing big congregations, socially gathering, et cetera, you give viruses the opportunity to spread. And here in the United States, of course, we value our liberties and our freedoms. And a lot of people are very, don't want to give that up. And they're willing to take the risk because they look at the stats and like, listen, it's not going to happen to me. But it happens to a lot of people. And it's very, very difficult when you lose a loved one to a disease like this.
Okay, I appreciate you visiting Infection Control today, Dr. Regan. Anything you'd like to add that I neglected to ask that you think is pertinent?
No, I just think this is a time really in the modern history which we haven't experienced before in the past. It's something which is very frightening. It's also something that historically, you go back to the 1918 influenza pandemic, we knew it was going to happen eventually, and here it is. And so, unfortunately, we thought we were more prepared than we are, and this has shown us our weaknesses. This also won't be the last one. And so we, again, have to look at how we are responding as a country, again, on the three fronts of fighting infectious disease I mentioned, and look to see how we can improve it for the future. Because invariably, there's going to be another virus, or you get antibacterial resistance, which is also a big problem. And so overall, we need to revamp the way in which we're doing testing, obviously for this pandemic, but also for other issues like antimicrobial resistance. I mean, these are big, big issues. They require policy changes from the government. And I certainly hope that what comes out of this horrible pandemic is better policies that will allow countries like the United States and across the world to better respond to infectious diseases, because often we do not give them credit, and it's to our folly. And so we need to do better.
Dr. Jack Regan, thank you for visiting Infection Control today.
Thank you so much for having me. Appreciate it.